All right, well, it's great to be here today. This group for HESI reaches all the way back to 2001. So we've been doing this for quite a while. Even though the context of what we're doing, which is looking at novel proteins and for the most part thinking about them in the GMO crop ag biotech field, is relatively new. And in fact, you really can just go back 20 years where those first generation products were registered, where the safety assessments were done, and they were put in the marketplace. And then shortly thereafter, some of the concepts I'm going to tell you about today were formalized, because there really wasn't the concept of what would make a good characterization panel in terms of allergy and allergy safety. For novel proteins, think about it in terms, we say novel, and some of the regulatory agencies around the world would describe novel as being one amino acid different than some native protein that maybe we're already exposed to. But for all practical purposes, the proteins that would be in these GMO products are just transferred. So a novel use, but not necessarily novel in the way we think about them for allergy safety. Part of the setup here and why we do the work is the fact that we're dealing with food. So, of course, in ag biotech, when you say customer, the product being sold is to a farmer. They're the customer. They're the marketplace customer. For all practical purposes, though, if you ask somebody about GMOs, they're thinking of themselves as a consumer. So why are we looking at this? Well, there's a global need for the food increase. The genetically modified crops, in terms of the increase in productivity, is a big part of that. What the PATC does in terms of addressing allergy, there's a certain expectation that you're going to address allergy because food is emotional and allergy has become more emotional. I'm sure just about everybody is aware how much more focus there seems to be on what people think are allergens and how allergic they may personally be. And you kind of wrap up the GMO emotion into that, and there's just an expectation that drives down through the regulatory agencies that ag biotech is going to do some very prescribed things for allergy safety. So the PATC is, I kind of think of it almost as a dual mission. We're kind of issues management, so there might be a regulatory agency in the world that really has what they think is a pressing matter in terms of allergy safety and the biotech novel proteins. But the reason that we exist is to look at basic allergy science, because allergy science isn't nearly as mature as, say, toxicology. This group, like I said, since 2001, I've been with the group since 2005, and in the immortal words of a colleague, I think this allergenicity thing will take about 20% of your time. It doesn't take 20% of my time, so it takes a lot more, and it's just because those expectations I talked about seem to be increasing almost every year. We've tried really hard, and I think we've done a good job with the tripartite aspect for at least 10 years. So, of course, we've got the industry folks, but we've got some really dedicated academic folks. Dr. Van Ree and Lars Paulsen from Copenhagen participate and travel around the world for us, and we'll go give talks even if the rest of us aren't able to go. We've had a fantastic manager, Nancy Dorr. I can't tell you how much we miss her. We most recently were in Kenya, and if you can imagine the logistics in setting up and going halfway around the world to work with Dr. Tanui and be there and participate in a workshop and think about a work product, she really heads that in a way that's almost unimaginable sometimes. The tripartite part there includes folks from the EPA, John Cowles, who some of you I'm sure know, really are dedicated and have been with us for some time. So a little bit about what we're doing. Because we don't have a lot of good definition around what makes a protein an allergen, that's where the basic science part comes in. And even though there aren't that many allergens in the world, and they don't come from that many source organisms. So to give you an idea, 65% of the known allergens come really from four main plant sources, for instance. But they're diverse in the sense that, the peanut allergens, for instance, of which there are more than one, aren't probably acting, and they don't act in exactly the same way as the kind of allergens or the proteins that are in, say, shrimp that give you shellfish allergy. So I'll talk a little bit more about that later, but that's where the basic science is. We're still at this stage trying to figure out that piece. Since we don't have a definitive test in the way you might think of a toxicity test, we kind of promote the weight of evidence approach. We've got several characterization assays to look at the biophysical similarity between, say, a novel protein and allergens, and we look at all those together, and then that kind of becomes the hazard and risk assessment. And then I'd say, one of the strengths of the PATC is the communicating piece. Everybody understands that when we go do our workshop, whether it be in China or Europe, that we let the speakers know that we have an expectation there's going to be some kind of written word product. So we've got a really strong publication history that is a lot of fun to do because it actually drives really strong talks if the speakers know they're expected to put something down on paper. What are the health risks? I told you there's a lot of expectations that the industry itself is going to look at allergy. Well, 20 years ago, we didn't even have a database of known allergens. And a lot of the allergens that we know of today hadn't been described down to the level of protein sequence anyway. But the concept was, well, there would be risk with a food, so you definitely probably were not thinking about taking a so-called novel protein from peanut and transferring it to corn. Because if you haven't described all the allergens yet, there could be a hidden one in peanut that you didn't know about, moving it to corn. That's much less so the case today. Well, I want you to think about organism as a risk factor, because it was definitely deemed a risk factor 20 years ago, less so today since we have good ability to describe exactly what's being moved. Something I don't think was really conceived of much 15 years ago, even though there was a little bit of regulatory work around this, but that would be, has that transformation process that made this novel protein able to be expressed in the crop, was there some risk there that it would increase known allergens? 20 years ago, we didn't have a good feel that there was anything such as corn allergy, but definitely soybean was perceived as one of the big 8 allergenic foods. So it's got endogenous allergens. Well, you can imagine that there's an underlying bias that, yes, GMOs definitely have some kind of unintended effect. Remember that word, unintended effect. And so starting about 7 years ago, the expectation was you're going to measure the soybean allergens and do it quantitatively, which had never been done before. So we've got some work that we've actually wrapped up in that area. And then, of course, the trickiest one is, can a novel protein become a new allergen? So I mentioned, the definition of novel may not be perfectly accurate, but it's really important to know who you are as a novel protein. If you're an EPS protein in that first generation Roundup Ready corn, we're exposed to lots of EPS-PSs. So the toxicology person well understands that if you have a good mode of action and you're ubiquitous to a certain extent and your exposure's already taking place and it's safe, probably not a risk. But you still get this question about, is this novel protein something I really understand? And could it become an allergen once it gets exposed when you do market that? The weight of evidence approach that I mentioned, it's a holistic approach. I'll list the things that we go through, so a panel approach. And we're doing that because we don't have that one specific type of test for allergy. And a lot of what the PATC does is think about that front end part, that screening or characterization work, but also what would further testing look like? So kind of that toxicology tiered approach, I'm going to do some screening work, and if I see something provocative in terms of adverse effects or a lack of safety, there might be some more definitive testing. That's certainly the case in allergy, because even looking for antibody against an allergen and a familiar allergen like peanut, if I take that serum from your blood, look at the antibodies to specific proteins, If I do that from a population perspective, it's not fully predictive. Seems like it should be, but it's not. So we think about what further testing would look like. So part of the goal today is, to give this some context, because the context has been around for a while, but the questions keep bubbling up to the surface depending on what the topic is, even now that we've been into this for quite some time. So I'll walk through an assessment. I'll tell you about some of the highlights with research, kind of one of the things we've completed, one of the things we're in the middle of, and one of the things, using some very new research. And then, of course, I'll talk about the outreaches, which is how people see us and see our work. So thinking about those characterization assays, I mentioned the source organism is really important. We now have databases of allergen sequences down to the protein sequence. So we can ask, is this novel protein similar, identical? And so we do a lot of that from a screening perspective. We're talking about food generally. So we know that if protein's susceptible to gastric pepsin, it's unlikely to reach the gut immune system. So that speaks to exposure. So we're going to look at that. 15 years ago, this concept that certain proteins were glycosylated, and that it was actually the glycosylated moiety that was part of allergy risk, instituted some screening for looking at whether the protein in the new crop plant is glycosylated. And of course, it's food going through processing almost always, certainly for soybean, although soybeans can be eaten directly, but we're talking about field crops that go into processed foods. So is exposure likely after heat and processing? Is abundance high? Is exposure the kind that would look similar to peanut, where the proteins in there are food storage proteins at very high concentrations? And does the protein have any history of unsafe exposure? So we're going to, these types of characterization studies go in one or the other column, so we can say something about the evidence holistically. So that is the future for predicting allergy risk. We can't truly predict, but we certainly can characterize things pretty well today, certainly all the way down to a molecular level, now all the way up. Bioinformatics, the concept that you can take a sequence and compare it to many other sequences, say in an allergen database, is something that's matured quite a bit in the last 10 years. Serum screening. Serum screening is one of those things that I mentioned, the follow-up testing. So say I screen a novel protein against my allergen database and it looked similar but not exact. In the regulatory guidance, that would drive some kind of follow-up study, and that would look like serology or serum screening. I'm going to take serum from people that are allergic to that organism that produces that allergen and see if that serum can bind with its antibody to my novel protein. That would be serum screening. Proteomics, I'll talk about determining epitopes, so the actual discrete parts of a protein that make it allergenic. That's part of bioinformatics, and we pay attention to that. And then, of course, the holy grail on here is, could you ever have an animal model? So something that would represent the systemic exposure of your novel protein, and you would maybe have a collection of endpoints that you could measure that you know are consistent with allergens. That's really tough because allergens only affect a discrete part of the population. So trying to think of an animal, whether it's inbred or outbred, as a model system that somehow is going to represent a very small fraction of the human population against only one allergen is a really tough thing, and it hasn't happened yet. You can get mice to respond really well to ovalbumin with an adjuvant, but I can't line up my top 10 allergens, from a prevalence standpoint and get that mouse to respond to all 10. So thinking about cell-based assays and in vitro assays to represent one or more parameters that might, that you could maybe use to screen with. But animal models still sits out there as that holy grail for us. So what are we talking about in terms of the mechanistic part? There are four basic types of allergy, but the one we pay attention to is really the type 1 acute hypersensitivity, the kind of allergy that you would get if you're allergic to peanut. You'll respond within minutes. Several different types of symptoms that go with that, but the mechanism is you're going to have repeated exposure to an allergen such that you're going to get some memory in the form of IgE antibody. That antibody sits in your system as the memory mechanism. Upon subsequent exposure, you're going to get the symptoms by way of the mediators produced by those mast cells, basophils. So that's the type we think about. I'm going to a conference in a week where EFSA is asking for input on non-IgE mediated allergy. So the kind of allergy that would be associated with gluten enteropathy, wheat, barley, rye, celiac disease oriented. They're asking, is there something special we should do for that type of allergy? So we'll be discussing that. So one of the areas of research for us was we were getting in feedback in, say, the 2006, 2007 time frame, and this is when Omics was already very popular in certain scientific areas, but hadn't quite bubbled up in the area of allergy work or allergy safety. And so to kind of get ahead of that, we knew there was this risk that some of the regulatory agencies would say, well, we know you can have panels of 100 genes or 100 proteins. Why don't you put your novel protein in a mouse and just look at all 100 and see what you see? Well, you could from an academic standpoint, but think about that in terms of safety testing, not so straightforward. So we knew that omics could possibly apply to this question I mentioned to you about endogenous allergens. So the underlying premise that the GMO technology might increase known allergens. Well, for soybean, there's upwards of 20 plus sequences that may have some clinical evidence. So to think about quantitatively measuring 20 or more sequences so that you could compare that GMO variety against this non-transgenic comparator was a difficult concept. And the serology that I mentioned, where maybe you would go out and find soybean allergic people, well, serum for one person can tell you something about how that one person's responding, but as a reagent to measure how much is being expressed doesn't work so well, because the next person and the next person each have a different panel, so to speak, of those IgE antibodies. And so as a reagent, that doesn't work so well. So proteomics, one of the things that was new at the time was mass spectrometry. So mass spectrometry, looking down to fractions of a protein sequence to describe exactly what protein you're looking at, but do it quantitatively. The hardware for doing that had not matured up until about 2007, 2008. And so that was the way to basically do targeted proteomics. And we could offer that if the agencies felt really strong about this, measuring endogenous allergen, here was an analytical way to do it rather than the serum-based approach. And it is the case that it's just extremely difficult to even get soybean allergy or serum. There just aren't that many people, especially adults, that are clinically allergic to soybean. You typically have to go after children, and it's tough to get enough serum to even do that kind of work. So we worked with Jay Thiel at the University of Missouri. He's a leader in the field and was already a leader in the field at soybean proteomics to start with. And that matured in the form of an established assay for both extracting proteins from soybean, but also using mass spectrometry. We've got several papers with him, and that's turned into one of the standards. Certainly you can develop, say, ELISAs to look at soybean allergens, and people will do that too. But this was novel and kind of pushed the envelope even for the field of mass spectrometry. So that was really fun to work on. The context for doing the endogenous allergy, though, one of the things we worked really hard at was that we knew the agencies, which are ultimately the customers for this, that they would just want you to compare the GMO variety to its non-transgenic comparator. But we said, well, this is a new technology, certainly in the form of it being quantitative. That's not the proper risk context. The risk context is, if I want to measure this new allergen, new meaning cunitrypsin inhibitor, hadn't been measured quantitatively before, then the right context is to understand natural variability. So how do soybeans normally express it? because it's likely to be the case that if I take GMO and its comparator, they're the same variety, and if you plant them in the exact same site location in the same year, there's going to be a very minor difference because there is no evidence that the transformation process would even cause a difference to start with. So you're setting, the setup was for a bad experiment to somehow compare GMO versus a comparator without understanding this type of variation, and this is several different varieties of soybean and just asking what's the natural variability from one site location. You can see, relatively speaking, quite a lot of variation. Then you can ask, is my difference between the GMO and the comparator fit within this, or does it lie outside? And so we've taken a long look at this, and these types of studies are ongoing. And here's another allergen, glyam-4 from soybean, and the range is 19-fold over the set of samples that were taken. And you see a list of some other allergens there from soybean. And so this fold range is the natural variability, and we've just tried to promote the idea that the best question to ask is, are you measuring an important difference when you compare the GMO variety, and does it fall within this range? Because unless it falls outside this range, you're probably not able to describe any kind of risk, real risk for the people consuming it. So that, like I said, is maturity. Even though these types of studies are ongoing, this type of work in the form of a formal study is an expectation, for the GMO varieties being registered. And the end story here is that we want to let them know that in terms of a commodity crop, that just by variety, the non-allergenic population safely consumes this extremely dynamic range. And so I mentioned in part of the characterization studies we do that there's this pepsin susceptibility. We call it the simulated gastric fluid. And the origin of this test, and this is a plate-based type test, and that's important because the original idea was that we knew that some allergens were very stable, and a lot of the peanut ones are, for instance. Stable meaning something on the order of more than 10 minutes in this plate-type assay. Pepsin put in with the purified novel protein, pepsin put in with the purified allergen, and you could look at how long it took for the protein to be essentially digested. And so that had been going on for many years, up until there was some rewrite of the European guidelines, and the draft version being in 2010, and the draft language in there was there could be a possibility to provide a more physiologically relevant pepsin digestion assay to address novel protein safety. The concept is, like I mentioned, if pepsin digests your novel protein in a very short amount of time, then you might not or probably don't have exposure to the gut immune system. But this creates a lot of confusion because that test is sometimes called a model. You're using an enzyme that we know is in the stomach at a certain pH. It must be really simple to put that in a 96-well plate and add your purified protein. Well, it's just a test and it's just a screening test, but trying to communicate that it's not a model of the very complex action your stomach's performing to digest food, and that those two aren't the same, can be a challenge. So this language showed up in this draft guidance, and the idea, and again, this is not real clear because it's difficult to communicate with European authorities, is that this more physiologically relevant digestion assay should account for the fact, for instance, that your stomach doesn't have a perfectly constant pH of 2.0. So we're in the middle of this, meaning we knew this language existed in 2010, and the idea that PATC would take this on in a laboratory setting and develop what could or would be a new protocol, but at least look at it from an academic perspective and see was it even a viable concept. And just a quick schematic of what that looks like. Gastric phase and duodenal phase, you would expose the purified protein with pepsin at either no ratio or a 10-to-1 for different time periods, and you address that for both pepsin, but you also address it for the duodenal phase where you have trypsin and chymotrypsin, so kind of a combination assay to address the full exposure panorama for that protein going through the initial part of the gut digestion. So we're in the middle of this, and I think, to me, the most important part is we said, we know there are allergens that belong to structural classes where the rest of the items or the other proteins in that structural class aren't allergens. So a good example is tropomyosin. So muscle protein and the ones in shrimp will cause shrimp allergy. The one in other organisms will not. And so thinking of it in a paired, so even though we don't know specifically why shrimp tropomyosin is an allergen, but we could at least have a control for that structural class of protein and set this up using pairs of proteins. And so that's what we're doing here, and the idea is to look at this range of pH values over these range of time courses to understand whether a new protocol even makes sense. The last, or one of the last I'll talk about here in terms of a platform we're looking at. We have a collaboration with the Japan National Institute of Health Sciences. Dr. Rieko Toshima, one of the regulators there that we've worked with for some time. I mentioned looking at the endogenous allergens. Well, this is another platform other than mass spectrometry. Electrophoretic concept, the idea is you separate proteins to look at single proteins, and this was part of their interest because that's the platform that they felt could do that kind of work. And it's difficult to make gel electrophoresis quantitative, so the idea was to test that concept and do it in a ring trial setting. And the plan was to do that and see if the protocols themselves could really be validated. So 5 independent academic laboratories taking a look at that platform and focusing on rice, with Japan focusing on rice in terms of the quantity that is in their consumers' diet. And so using this 2D DIGE platform, they took this protocol, and a very complicated protocol at that, with the overall goal to turn that into a repeatable, simple, and distributable protocol. And I don't think I probably have to walk you through this to describe how complex this can get in a hurry, but the idea here is you're not linearly separating proteins. You're going to actually separate them in 2 axes, and a dot can be a single protein. Just I've highlighted a couple up there, but trying to graphically, so to speak, circle a protein and do that repeatedly is very challenging. But they did get the end of their project. They've gotten to the point where they can put a manuscript together, but I just wanted to schematically show you how difficult that has been just for that platform. So one of the things we've just newly taken on is something that doesn't perfectly fit with allergy, but there was that IFBIC committee that focused on toxins and asking whether we could describe toxins the way we try to describe proteins, and down to the sequence level to think about doing the bioinformatic piece. So if I've got a novel protein, could I do the same thing where I compare it sequence-wise to my allergen database? Could we maybe someday do that, create a protein toxin database? So we brought that under PATC because a lot of the same folks work in this area as they do with allergy, and within particular the bioinformatics. And so focusing on the bioinformatics, here are just some of the goals, and kind of the end game here would be a curated protein toxin database like what we have for allergy. So we're managing this as a separate work stream. This is kind of novel for the PATC because otherwise all the participants that we normally have, say on our monthly calls, focus on that. Just a select set of folks said they wanted to work on this, and so we're managing this just a little bit differently. And one of the things I'm going to end with that represents kind of an emerging technology that we're taking a look at is going to probably sound real familiar to everyone, but this is using a genomic our transcriptional screening panel. So the idea here is to take something that's been developed to a certain point in a lot of different scientific areas and ask, can we have a panel of genes that we know would at least have some relevance to allergy response at the cellular level, take a cell line, a dendritic cell line that we know is important for the beginning sensitization response phase, and ask if we can put some purified protein, some control proteins and some test allergen proteins in this cell system. Can we look at the up or down transcription of those genes and ask, can we tell the difference between a known sensitizing allergen and a control? And so just a quadrant schematic of whether there's separation between the known allergens and the known non-allergens. And these graphs, the allergens are above the horizontal axis and the non-allergens are below. And this is some preliminary work that was sponsored by Novozymes to take a look at airway sensitizing proteins, since that's kind of the business they're in terms of purifying them large scale and having to pay attention to occupational exposure through the airway. And so the PATC is going to supported a proposal for this, and the GARD assay itself is genomic allergy rapid detection, and asking, can we take those paired proteins that are in this pepsin assay design that we've got, put them in this, and see if we can learn something there in terms of separating allergens from non-allergens. So again, emerging research for us. It's just starting here in August. How am I doing on time? Okay.
Yep, that's fine.
Yeah. So I'll end with outreach. I like this part of what we do for PATC because it's so dynamic. Every year's budget includes outreach to all these places, and everything from 2015 all the way back to 2001. And Dr. Tanui that's going to talk to you today right after me, we were able to coordinate, and we had worked on this for quite some time. We coordinated with a workshop that had been planned in Kenya, and we made that happen last August. And so that's, to reach out to, for us, that was a new part of the world to take a look at. But this just kind of, without me walking through all these, gives you a feel for all the places we've been. So kind of exciting in terms of where we can take our message, because we're dealing with a lot of different regulatory agencies in the world, a lot of different flavors of what they think they should have to do for their populations, and it's not all the same. So one of the big questions, you heard me mention the words unintended effects. There's kind of this underlying bias that there must be an unintended effect of making a transformed crop product. That kind of bubbles to the surface in the form of both the endogenous allergens for this group, but overall crop composition, for which there's a lot of analyses. And so we had a big workshop in Canada last year that turned from Canadian officials wanting a very small conference just to have a very close face-to-face into an international conference. And we got a really nice publication out of this that everybody worked really hard to make this happen. And it puts in one place a lot of the key references in this field, because it had really only been published on in terms of data heavily in the last 5 years or so. I mentioned the workshop in Nairobi, Kenya, and that was a lot of fun, and we interacted with a lot of different groups, so we were there over the course of a week, so it was not just a one day type of thing. And so that worked out really well that way. And when we do that, we take a lot of the pieces and parts that you've seen today from me, and we try to give it that context that I started with today. What kinds of questions can you ask, and what does risk really mean? Because we don't want to just be a hazard assessment. It's got to include knowledge about history of safe exposure and exposure itself. And that's not always well put together in terms of the regulatory language that some of the countries in the world are just now beginning to put together. And so we do that, and then we took that Ottawa conference, and we're at OECD in April. So we had folks from EFSA and the EU at the Ottawa conference, and they said, can we get this to the regulators that weren't able to travel? So OECD opened their doors for a satellite meeting, which is hard to get with them, but they let us have that, so we were in the same room where they're holding their annual spring conference. And so we could take some of these focus messages, like on the endogenous allergy assessment and consequences of unintended effects, which was important for us to be there. I'll wrap up with high-impact publications. I told somebody the other day what these numbers were, and he didn't believe me. I said, well, if I showed you the room that we talked to in China, it was more than 200 folks, but I would say the majority of them were students. So all the regulators, representation of the different groups were there, but since they're also academic folks, they had their students come. And so I feel like a lot of those downloads are from all those students in an emerging part of the world in terms of a regulatory system, taking a look at the outcome of what they heard us talk about. So I thought that was pretty cool. It's hard to know what the real impact is, but at least you can see that there's activity in reaction to us being in a certain place. So I'll end with that, and I want to thank Cyril and Michelle for helping me put this together in short order, and a lot of fun to be here, so thanks. I biased the right side of the room or the left side of the room, sorry, but I like to walk like Cyril does.
Can we take a couple of questions and then?
Yeah. Been a long time, Dan.
Monsanto. I think we can talk to each other this week.
I think so.
We may be working on the same next week. Let's see what happens. So I've been following this issue for 16 years, and if you look at auto safety, one way you could die is being hit by a meteor while you're driving your car. And we don't have any safety programs in place to address that. And you can develop assay systems to develop meteor-assisted vehicles, but since nobody ever gets hurt, I'm not sure you could ever prove one system is better than the other. After watching this for 16 years, the safety assessments that we've undertaken haven't actually added any demonstrable value beyond the fact that we shouldn't add known allergens in to foods. We're looking now just endogenous allergenicity issues. We're up to 22 allergens in wheat. They want us to measure them all. We have no idea where those protein levels fall in regards to the overall stochastic dose response to the general population and whether changing these levels will matter one bit. It isn't clear that we can do anything about them because the odds that they are independently breedable, if you want to get them all to go down, are very slim. Many of them are functional proteins and can't be taken out at this point. Meanwhile, we know where the real problem is, which is existing food allergens that we can't engineer out at this point in time. And I guess my question, this is not to be critical of the great work that you've done, but to me, the key question for PATC isn't how do we do this better, it's how do we stop doing this? We've dug a hole. How do we stop digging and get out?
Yeah. Well, so that if I could show you one of our full-length presentations on endogenous allergy, which we presented once again at OECD in April, that's the very first thing we mentioned. That we can tell them we worked hard on doing the thing that they wanted done more quantitatively and better because the old way was not correct in any way, shape, or form. It was, but we start off with, why would you even do this in the first place? And then now this is expanded to, I mentioned this last part that we're doing with the unintended effects, and it's kind of the same question. And now that we can put the molecular biologists and the breeders in the same room while we're having a discussion, they will tell you that there is no evidence that you can relate the variation in allergen content to anything that's being done to move a protein into the GMO variety, and in fact, breeding itself creates more unintended consequences, even from a safety standpoint, than anything that's ever been done by moving just one known protein. So we always start out there. Yeah.
Simple question. I suspect I know the answer, but I'd be interested to hear why 2G geoelectric recess? This is a technology that's over 40 years old.
Yeah.
A lot of labs just jump straight past it into mass spectrometric proteomics because of the difficulties you've mentioned. So I just
Right.
know that is there any actual additional value to using straight mass spec?
Well, I will say as a platform, though, what's gotten better, and even today this gets better virtually every 6 months, is the optical detection equipment and the software that you need to use to draw those little circles. That's what's gotten better. But for us to work with it, what it actually was an opportunity to work directly with the Japanese regulators. So it was a favored platform by them. And because the regulators are also academics with their given labs, that happened to be one that they were focused on. And so it was a chance for us to say, well, if you thought you could get to the point of having a protocol, well, we can support the next step, which is ask, does the variation in the technique itself swamp the ability to even use it quantitatively? So it was all of that kind of wrapped together, yeah. Yeah.
Are you dealing with proteins added to food at all, so enzymes, or even do they matter?
Yeah, I mean, manufactured,
Enzymes manufactured, used in food processing.
yeah, purified proteins that add nutritional value, what have you. I mean, when we all think of novel proteins, we do think of those, but the regulators regulating the GMOs are typically they're not the same folks regulating those. So when we think about kind of these platforms, we're not typically looking at those as much as we are, the novel proteins. But it could be a novel protein from any source.
Okay, but do you think they matter? Are they likely to be allergenic? Are they likely to be problematic?
Oh, you mean
That's on.
Is it on? Oh, okay. Yeah. I mean, these enzymes that are added to food, are they likely to be allergenic? Are they likely to be problematic?
Well, I mean, if you took an enzyme that is an allergen, which there are not that many of, you would go through the same risk assessment. In the EU right now, they do manage that separately for the manufactured enzymes because they do get used a lot for food and feed, value-added ingredients. And so, yeah, if you took a known allergen, or what really is the higher worry is occupational exposure through the airway with some of these proteases. So they have a good feel for which proteases and enzymes are sensitizers, but as a food allergen, there's not much of a worry because there's going to be pepsin degradation of those enzymes. I don't know of a pepsin stable food ingredient enzyme that would be a worry the way we normally think of it. Yeah. Do you want that?
So I had a sort of an interesting question regarding pepsin and this might show a little bit of my ignorance because I don't know much about protein antigenicity and so on, but from your readings, have you looked and are you aware whether pepsin breakdown of proteins is a way to decrease antigenicity of some orally ingested proteins? The question that I ask this is because, coming from a pharmacy school setting and looking at things in terms of therapeutic use of other medications, proton pump inhibitors are now over the counter.
Okay.
They're highly abused, right?
Okay.
The pH of the stomach increases significantly, when you use PPIs, of course, and that has an impact on the activity of pepsin. From your readings and talking to people, improper or suboptimal activity of pepsin, could that be somewhat linked to a potential impairment of decreasing or erasing antigenicity of orally ingested proteins?
We think that's exactly why we've gotten this new regulatory language, because I think they're trying to recognize the fact that you could have those, or you could just have simple, good old Maalox that's being taken at the same time that you're eating an allergenic food, and that you might be running your test at pH 2, but in reality, the pH might start at 2 and then go up and cascade through a range of pHs, and that an allergen, the novel protein, might make its way. So we think that's where the question is coming from, but that's also part why we've set up the design to also include the duodenal phase, so that you've got the next set of enzymes in the cascade present in the assay format. But it is the case that pepsin operates in a really narrow range. And so we know that's why we usually try to give this context in the sense that the test, just to make that comparison, is not meant to be a model of what might happen in reality, because that pepsin test, the way we run it, is put in that weight of evidence context. So the idea is there no one assay could presumably fail you or
Yeah, I was just getting to the point where somebody's looking at use of antacids and propensity for food allergies, and whether there's a relationship between those two.
Yeah, I don't know. I mean, some of the studies you'll see when we look at animal models for allergy, we consider it a flaw that with some of these oral exposures, that one of the most difficult things to control is the pH, and so that in conjunction with the adjuvants they're trying to use, I'm sure the pH and the actual pepsin activity is being modified, but you don't know how. So yeah, it's a tough one to bridge the expectation that you've got a model to the expectation that you're just making a comparison. I don't know if I answered your question exactly.
Okay. Thank you, Dr. McClain. Were you going